The disorder we chose to research was achromatopsia. My name is Samantha Young.
I'm Charlene Madden.
And I'm Laura Wagner.
We picked this disorder because in this last section we went over a lot of colorblind genetics problems, so it was interesting to see a disorder that was a little bit more in-depth, but still related to what we were talking about. Achromatopsia is an autosomal recessive disorder. It can be categorized as a loss of cone cell function in the retina. Your cone cells are a type of photoreceptor in our retinas that are responsible for color vision. Some symptoms of achromatopsia are color blindness, photophobia, which is an extreme sensitivity to light, and nystagmus, which is an involuntary eye movement, also known as eye dancing. People with achromatopsia also have a severely reduced visual acuity, which is their sharpness of vision. Usually the affected have less than 20/200 vision. Achromatopsia is present in 1 in 30,000 births.
General signs of this disorder can be seen in an afflicted person in as early as 6 months of life. An extreme sensitivity to light and involuntary eye movements are two warning signs of this disorder. Currently, there is no known cure for this disorder, so the focus is on symptom management. Patients with this disorder generally avoid light as much as they can, as well as wear specialized glasses that reduce glares. There is a lot of variability in this disorder, so some patients can have incomplete achromatopsia, meaning that their vision will still function somewhat normally, in comparison with those with complete achromatopsia. For patients with this incomplete achromatopsia, there are special glasses that can help them distinguish between certain colors.
The inheritance pattern of achromatopsia is autosomal recessive. Some ethnic groups see a higher prevalence of disorder when they can trace their ancestry back to a specific geographical area, which may explain why it is increasingly prevalent in islanders living on the eastern Caroline Islands of Micronesia.
The CNGB3 gene is responsible for encoding for a protein that makes up the beta subunit of the cone photoreceptor CNG, which stands for cyclic nucleotide gated ion channel that's found in your retinal tissue. More specifically, these channels are found in the cones of your eyes, which are responsible for daylight vision. CNG channels allow for phototransduction to occur in cones. Phototransduction is a process where light is converted into electrical signals in your rod, cone, and photosensitive ganglion cells in your retina. In your cones, phototransduction occurs when the CNG channels allow cations to flow into the cells in the dark, but stops the flow when light enters the eye, altering the cone's electrical charge, which generates vision. A mutation in the CNGB3 gene, which accounts for 50% of achromatopsia cases, alters the structure of the CNG channels, which makes them non-functional and unable to perform phototransduction because the CNG channels will allow an influx of cations into the cell, which leads to apoptosis in cone cells.
The CNGB3 gene is located on chromosome 8 in position 21.3, which can be seen in the picture. The CNGB3 gene encodes for the S435 protein. This protein's normal function is to make up that beta subunit of the CNG channels within the retinal tissues of the eye. The mutation in the CNGB3 gene involves an amino acid substitution due to base pair deletions. The amino acid serine is replaced with the amino acid phenylalanine at amino acid position 435. This new protein is being referred to as S435F. The research article we chose was titled, "The B3 subunit of the cone cyclic nucleotide gated channel regulates the light responses of cones and contributes to the channel structural flexibility." The purpose of this experiment was to investigate the functional properties of cones lacking CNGB3, as well as the biochemical features of the CNG channels lacking the CNGB3, in order to better understand the role of the gene in the cones. In this experiment, mice were used to analyze the light response properties within their cones. The steps listed in the slide are directly from the journal article, with each step consisting of a paragraph of explanation of what occurred. Basically, the researchers acquired mice with different genotypes dominant in cone and rod function, as well as those not. They isolated membrane proteins and took cross-sections of the eyes in order to observe them with immunofluorescence microscopes. Electroretinograms were then used to measure light responses of the different genotypes. Based on the responses, they were able to conclude how the levels of CNGB3 affected those responses and analyze the function of the CNG channels.
By using Ganzfeld phototopic electroretinograms, they were able to study impaired light responses in CNGB3 mice. They recorded wave responses in mice, which allowed them to evaluate the direct effect of CNGB3 deletion on cone function. Overall, their data shows that there was a reduction of cone responses and cone-mediated secondary neuron responses in the absence of CNGB3. By using electroretinograms, they also recorded cone sensitivity, response kinetics, maximal response amplitude, and phototransduction adaptation. By comparing the cone-driven responses in wild type in the CNGB3 mice, it revealed a reduction in maximal cone response amplitude, lower sensitivity, and abnormally slow recovery kinetics. They also studied the altered background light adaptations in functional range in CNGB3 mice. The deletion of the CNGB3 dramatically altered the adaptation capacity of cones and compromised their ability to function in bright light.
As you can tell by the data shown in these graphs, mice deficient in the CNGB3 gene show an impaired light response, impaired cone light response, an altered background light adaptation, and decrease in functional range. All of these are also commonly present in patients with achromatopsia, reiterating that the CNGB3 gene plays an important role in causing this disorder.
There is still no known treatment to cure achromatopsia. There are only ways to manage it, for example, the specialized glasses we mentioned in the previous slides. As shown in the research article, there is investigations being done on the functional properties of cones lacking CNGB3 and the biochemical features of CNG channel complexes lacking CNGB3. This allows us for further understanding of the role of CNGB3 in cones. This is a helpful insight and hopefully can be further utilized in order to expand the overall knowledge on this disorder and eventually help us find cures for it.
Normally, patients with achromatopsia experience a loss in cone function, and their rods are responsible to keep their vision as normal as possible. In recent trends of research, people are focusing on mutations that cause rod dysfunction as well. In the research article, CNGB3 mutations cause severe rod dysfunction. Their purpose was to show the results of an electroretinograph in two siblings with CNGB3-associated achromatopsia. In their research, they wanted to focus on patients who already had achromatopsia with cone dysfunction and wanted to see the effects of a mutation that also caused rod dysfunction in achromatopsia.
Thank you for listening. Here are our references.